we doing so far? 3-domain system? And I couldn't do it, but if you see the 5 kingdoms on your kids' wall in middle school, you should absolutely call them out on that. Because that is not current, not even close. All right, so here's a picture of the Wu's tree. He's got a great name. Remember we talked about Luca, the last universal common ancestor? So here's Luca, and these are all the bacteria. And we know that there's way more bacteria than everything else, right, in terms of diversity. And then Luca evolved into a new common ancestor, but that's common only to the eukaryotes, and then all of the archaea. Right? Kind of cool. I think it's so neat that, I mean, I'm old, but not that old. I mean, just in my lifetime, how much has changed in terms of what we know? And that's one of the really fun things I like about teaching biology. I've actually taught chemistry too, because my degree is in chemistry, but my bachelor's degree is in chemistry. But I always say those electrons fill the orbitals the same way every semester, right? But when you think about biology, it's constantly changing. It's so much fun. Or being updated or refined, or understanding being refined. All right, so I changed the study guide question. If you printed out the study guides already, I apologize, but this study guide question has been problematic for a while, so I updated it. And this is what would show up on a test or a quiz. The old one that was in there, if you printed them out, it's gone. This is the new one. So, but let's go over it. All right?
So this one?
I don't, so, that one's not working either? We'll have to fiddle with it. What was the question? Just that one question. I didn't change any of the other questions, just the one question. But this particular question, I think it said to use Wikipedia and try to find characteristics of the 3 different domains. And that's actually not that useful. This is more important. So what are the 3 domains of life? What did we say they were? Bacteria, archaea, eukaryotes, right? What molecule did Carl Woese use to divide all of life up into these 3 domains? Ribosomal RNA, that's the molecular clock, right? And which domain is believed to be the precursor to eukaryotic organisms? The archaea, right? The archaea evolved first, and then the bacteria were here first, right? And then archaea branched off, and then they were the precursor to eukaryotes. Make sense? All right, this is kind of a cool little video, so let's watch it.
Classifying living things is kind of like Pokemon. It appeals to our instinct to collect and identify. And the fact that we have a classification system is super useful for scientists. I mean, it shows how every type of organism we know is connected to all the others. We've mapped out all of life in one giant chart. It's amazing when you think about it. But deciding how to classify a new type of organism often isn't simple, especially when it comes to assigning it a species. In some cases, it's obvious when 2 things should be separate species. A jellyfish clearly is very different from a jaguar. But the more similar organisms get, the harder it can be to sort them. There's no one rule that will always tell you whether 2 organisms should be in the same species. So different taxonomists use different rules in different situations. The most widespread definition of a species is called the biological species concept, or BSC. It was popularized by evolutionary biologist Ernst Mayr in 1942, and he continued to refine the idea over the next several decades. According to the BSC, organisms are part of the same species if they can produce offspring that can also breed. If they can't produce fertile offspring, that means they belong to separate species. The BSC hinges on the idea of reproductive isolation, where something separates 2 species to the point that they're not compatible anymore. That separation could be time, or geography, or behavior, or physiology. Alder and willow flycatchers used to be considered part of the same species because they made up in the wild, they look the same, and it seems like they should be able to breed. But ornithologists eventually realized they're totally different species. They sing different songs to attract different mates, so alder flycatchers never try to mate with willow flycatchers. They're reproductively isolated. A species is therefore basically a gene pool, one that doesn't overlap with the gene pools of other species. That's a good, rigorous scientific definition. It can be tested and observed in the wild. But there are lots of cases where it doesn't work, like with microorganisms. Bacteria, for example, don't reproduce sexually. They can share genetic material with each other, but it's not part of the reproductive process. So reproductive isolation is totally irrelevant, and a scientist looking to classify a new species of bacteria can't rely on the BSC. Instead, they might look at genetics, the shape of the microorganisms and the way they grow, and at their evolutionary relationships to other organisms. You also can't use the BSC to classify fossils, since you can't exactly observe the mating behavior of a skeleton preserved in rock. Sometimes you get unbelievably lucky and two specimens are preserved while they're mating, but that's way too rare to be definitive about which animals mated with which other ones. That's why paleontologists usually use morphology, or the physical characteristics of a creature to classify it. Then there's the issue of hybrids, where creatures that should be part of separate species mate. When two species hybridize, there are a number of things that can happen. Often, even if they produce offspring, the offspring are sterile. That's what happens when you cross a female horse and a male donkey, for example. The offspring are mules, and mules can't breed. But sometimes, two species can produce fertile offspring. And biologists have found that animal species hybridize way more often than we used to think. If the offspring go on to mate with their parental species, it's called introgression. It provides a means for gene flow between two species, meaning their gene pools aren't totally isolated. So does that mean that you have to revoke their status as separate species? Scientists haven't settled that question yet, but some think the barriers between species are much leakier than the biological species concept allows. Sometimes this leakiness can lead to two groups being classified as subspecies within a single species. Subspecies give you a little wiggle room. Sometimes organisms overlap geographically and can share genes. In cases like these, subspecies means we know that they can share genes. It's just that they usually don't. Then there are ring species. In California, for example, there's a species of salamander that lives in overlapping populations of subspecies on either side of the San Joaquin Valley in a big elongated ring. Those subspecies can all interbreed except for two. At the southern part of the ring, two subspecies can't interbreed. So according to the BSC, those two subspecies should actually be separate species. But even though they can't interbreed, genes can still flow between them through the other subspecies. Where does that put them? You can't answer that question with the BSC. So having a hard and fast definition of species that you can test and demonstrate is useful, like in conservation when you're looking to protect a species and its habitat, but the biological species concept isn't an absolute rule that scientists are bound to.
So I just like to show that because it just shows you how hard it is, right, to create rules in science that you can follow. And that ring species example is great. And it's nice that he talked about the fact that you can't think about, we talk about bacteria as a species, but in many ways it doesn't make sense to think about bacteria that way. All right, so the classic classification of bacteria is based on phenotypic traits. So we said their shape, how they stain, the gas they produce, their physiological needs, etc. A lot of challenges that we touched on last time. There's not really a fossil record for bacteria. The definition of species, as I just showed you that video, doesn't work well for prokaryotes. And prokaryotes can actually look really similar to each other, and then it turns out when you sequence their DNA, they're totally different. So they're tiny, as those of you in the morning lab saw today. It is really hard to get a lot of information about how similar they are by looking at them. The other thing about prokaryotes is they regularly undergo something called lateral gene transfer. That's where a bacteria that has a helpful gene just hands it to another bacteria and says, here, have some of my DNA. It's not, you give your DNA to your children, right? You can't just pass your DNA off to someone in the same generation. The bacteria can do that. They can actually hand off their DNA to another bacteria. And that's called lateral gene transfer because it's happening laterally or horizontally in the same generation of organisms. We're going to talk about this a lot more as the semester goes on. So nowadays, we classify microbes using genotyping. So we're going to sequence the DNA, we're going to look at the ribosomal RNA, and we're going to even look at the proteins that are encoded by that DNA to try to group bacteria into evolutionarily related organisms. And that is really the only way we can do it. The species concept is of limited value. Just a little bit of boring terminology for you. Microorganisms, sometimes people call them microbes. If people are doing a lot of genetic engineering, we tend to call them bugs. They're not necessarily germs, though, because most microbes are not pathogenic. Only a very small fraction of microbes are actually pathogens. Most of them are useful or essential for life on the planet. Think about those bacteria that produce oxygen. Would you want to disinfect them and get rid of them? So the answer is no, because you need that oxygen to breathe, right? What about the bacteria that help fix nitrogen so crops can grow and you can have food? You kind of like those guys to stick around too, right? So what I'm trying to impress upon you is that the vast majority of bacteria fall into that category. And even though we're going to spend the entire semester talking about the pathogens, in as much as you can remember that actually most of them do good things and we need them to be here, we need to protect their environment and their ecosystem so they can be successful, it's a good thing to keep that in mind. When people talk about probiotics and ingesting bacteria to stay healthier, they're really thinking about keeping your own internal ecosystems healthy because that's actually good for your health too. So taking antibiotics is really important if you're going to die from an infection, but actually not good for you unless that's your only option. Because you want to keep the bacteria that are living in you and on you. You're an ecosystem. It's a community. It's not just you, but they're helping you they're helping you stay healthy as much as you need to provide a home for them. All right. Types of microbes, we're going to focus on bacteria, but there are viruses, there are yeast. We look at some protozoa, they're microorganisms, they're small. And we're actually going to look at this semester towards the end at some worms, because even though most worms are big enough to see with the naked eye, in their larval stages they can be microscopic. And oftentimes those larval stages can get into people and make them sick. So that's pretty interesting too. You should have seen this in the homework. When we name a bacteria, we think about the genus and the species. So Escherichia coli, Escherichia is the genus, coli is the species. And we underline or italicize the name. So I've italicized it here, but if you're handwriting on a quiz, you would underline Escherichia coli. The genus is capitalized, the species is not. It is acceptable to abbreviate the genus, so E. coli. So this would be S. aureus, right? Staphylococcus aureus. These are some really common. Pseudomonas aeruginosa. It's actually a pathogen of burn patient that lives in the soil. Bacillus subtilis is another soil microbe. Probably you can find some between your toes. We swap. Agrobacteria, tumefaciens, Mycobacterium leprae. This causes leprosy. So lots of blue lives. All right. So hopefully you're comfortable with the protocols for naming. When you think about bacteria, you kind of think about like a dog, right? All dogs can breed with each other, but there's different breeds of dogs. So bacteria are a little bit like, they can't all breed with each other because they don't breed, they undergo binary fission. But you can kind of think of different bacteria as, so within E. coli there are different strains. So E. coli, most of the E. coli is, you have it in your gut now, it's a commensal, it's doing good things for you. But O157H7 actually produces a toxin, and it's a fairly serious form of food poisoning. That's why you have to cook ground beef thoroughly all the way through, because we worry about E. coli O157H7. So it's all one species, but there's different strains, and it's usually because they have different genes that allow them to have some unique characteristics within the species. We don't use any pathogens in our lab. You're probably bringing more pathogens in that are in the lab, already in you and on you. We always buy them from the ATCC, and they're ones that are non-pathogenic. That doesn't mean you can't get sick from them. If you cut yourself and pour them into the wound, they'll still grow and eat you, right? You can get an infection, but they're not ones that just cause human disease for no reason. Okay, so we talked about this already. Okay, how are we doing? Yeah? You want to talk about Lyme disease and then do macromolecules? Is that a good, kind of a good, break it up a little? Okay, let's do that. So, disease of the day. I'm letting people give you a chance to get organized here. We're going back and forth between 3 slideshows. You know what you're thinking. You're saying, wait a second, that's not a bacteria. That's a tick. What do we say it's called when an insect transmits a disease? The insect is a vector, right? It's a vector for the disease. Okay, so Lyme disease is caused by a bacteria called Borrelia burgdorferi, and it's transmitted by ticks. So first thing I'm going to do is show you a little video. It's a zoonosis because it's also an animal, so usually if the ticks are on deer and they're on mice, They actually go back and forth between both. And so you need those organisms to be present for the ticks to end up with the right larval stages of, I'm saying that wrong. It's actually a larval stage of the tick, I believe, that bites you and transmits the disease. So animals are a reservoir, the mice the reservoir, and the vector is the tick. And actually there's an article here about Lyme disease, but what I'm going to focus on here is this video. This is pretty good.
There's been a lot of talk about infectious diseases from animals, like West Nile, swine flu, bird flu. So today, we're going to talk about one that's been around for a long time. Lyme disease. So let's put this in perspective. West Nile, which has gotten a lot of press lately, has about 2,000 cases per year, while Lyme disease has up to 35,000 confirmed. And it's estimated that only 10% of people report that they have Lyme disease. So that means that there could be 350,000 people with Lyme disease, a disease that if it goes untreated can cause permanent arthritic conditions and brain damage.
Well, if you find yourself with one of these, you better get it checked.
Turns out he's got Lyme disease. We're talking about a disease caused by ticks, an arachnid parasite that lives in trees and grass and is just waiting for mammals to walk by so it can latch on and have a little blood meal. Okay, there are a lot of types of ticks in the U.S., but only 3 of them are of major concern. And for Lyme disease, the big player is the black-legged tick. So what's the ecology of this disease? To help us understand this better, I went and talked to a disease ecologist and tick guy, Brian Allen.
So the ecology of Lyme disease is pretty interesting. So Lyme is caused by a bacterium, a spirochete, called Borrelia burgdorferi. The tick is not born infected with that pathogen. It actually has to acquire that pathogen from feeding on an infected host. So it's born as a larva, free from infection with the Lyme agent. It then takes one blood meal, molts into the next life stage, which is the nymph. The nymph takes one blood meal, molts into the next life stage, which is the adult. Adults take a blood meal, they mate, and they die. We think most of the human cases of Lyme disease are caused by that middle life stage, the nymph life stage. And so not only do we see a lot of cases every summer of Lyme disease, but they're increasing in number and they're increasing in distribution. Ticks actually breed on deer, and so how many deer you have in the area can influence how many ticks you have. And then the animals that are the carriers of the pathogen are things like small mammals, rodents, and things like that. And so large populations of small mammals contribute to high prevalence of the infectious agent.
So pretty much all of us live in a place with a whole lot of ticks, but don't freak out if you get bitten by a tick.
Every tick is carrying an infectious agent, and so you can be bitten by a tick and not be exposed to any kind of disease. Tick's not like a mosquito. A mosquito feeds really quickly, so it kind of takes a quick blood meal and flies away. A tick actually has to be on a host for a long time to take the full amount of blood that it wants to extract, often days. And so because of that, you may have actually a pretty big window of time to remove that tick before it can even begin to transmit a disease to you.
Let me just end by saying that understanding an ecosystem is really important for our health. And Lyme disease is a great example of that because you have a tick feeding on multiple hosts. It's all connected.
People get interested in ecology when they find out that there's a connection between ecology and their health. And so it's kind of fun to be one of the people who can spread that message that ecology matters.
You can watch more of our videos here.
Okay, so that's a really nice overall summary of Lyme disease and how it's spread. And we've actually talked a lot about ecology of hosts. We've talked about Zika virus and the mosquito traveling from Africa all the way to South America. We've talked about Ebola, where you have these animal reservoirs where there's these periodic spillover events into people. So, and the plague, we said, we looked at the ecology of the plague as well, where we showed that the plague bacterium actually cycles normally in rodents, but it can kind of get transmitted to dogs and cats, so people can get bitten by a rodent or bitten by a flea that bit a rodent. They can ingest. They can end up with the plague bacteria in their blood or in their lymph nodes. So Lyme disease is one that we worry about a little bit. It's on the rise. So there we go. So this is 82 all the way up to 2012. So this is the number of CDC reported cases, is in the dark. And this is the estimated total cases. So she was saying in that video that the number is actually a lot higher because a lot of people get this and get better. Their immune system takes care of it and they don't actually report that they've gotten it. So there's just some graphical pictures. So 2001 comparing it to 2015. And you can see that we are not free of Lyme disease in the Bay Area. It's here. It's not as prevalent as it is in the eastern United States. But I know in my neighborhood there's a lot of deer. I don't see the mice, but I'm pretty sure there's predating mice too. So there's the organisms that you need to have the full ecological system are definitely present. All right. So the common way people realize they have Lyme disease is that they get this erythema migrans rash. It's a bullseye rash. So it starts out and it ends up forming a sort of red bump in the middle and then a ring. It's very rarely itchy or painful, so you kind of have to see it. It can be kind of warm. It can appear on any area of the body, but it usually begins at the site of bite. And it begins days after the bite occurs. This is a picture. He said the bacterium was a spirochete, so that's actually a spiral-shaped organism. We're not going to see any of those in the lab, but this is a spirochete. So you can see how it's kind of got this spiral shape. The organism that causes syphilis is a spirochete too, Treponema palladium. Some of the pathogens, some spirochetes do make up some of the pathogens. All right, so that's Lyme disease. One of the important things about Lyme disease is getting it treated if you see it. Because usually people just end up with this rash, and if you take antibiotics, you get rid of the bacterium very effectively. However, if you continue to have the organism in you, as your immune system tries to fight it off, it can actually cause you joint pain. So usually these are all the result of your immune system. It can cause some Bell's palsy, temporary paralysis, but people tend to think of muscle and joint pain, nerve pain as a result of having. Some people refer to this as chronic Lyme disease, but there's no evidence that once you've been treated with antibiotics that you continue to have Lyme disease. That what's happened is that you have some damage from the Lyme disease done to your nerves or your joints, and that's continuing to cause the pain. So I had a student once in time, and I want to do a presentation because they would have to do presentations in there on chronic Lyme disease. And I'm like, yeah, there's no science behind that. So you got to keep it scientific. But if you go online, you'll see lots of people talking about chronic Lyme disease. The data's not on their side. How do we treat it? Well, it turns out that doxycycline, which is an antibiotic that actually prevents bacteria from synthesizing proteins, is a very effective treatment. Let's see, if you do this early enough, it's usually curative and people don't have any problems. If you have you've been infected for longer and you haven't been treated and it's really, this bacteria really in your system, it's been disseminated, then things like Rocephin, which is another one that's a pretty heavy-duty injectable antibiotic. They give it for people with pneumonia. That's the first choice. It's a big needle, painful. You don't want to get Rocephin if you don't have to. All right, post-treatment Lyme disease syndrome, not chronic Lyme disease. So these are people that are having pain after they've been treated. Lingering symptoms, I said joint pain being one of them, fatigue, damage to resulting tissues and the immune system probably causing that. Patients do get better, but it can take months for your body to recover from an infection like this. Okay, so that's our disease of the day. Any questions on Lyme disease? Yeah? A couple of, this spring, but the spring before, Matt Schweitzer, one of the micro teachers, he's like, I have a bullseye rash. Did you call the doctor? He went to the doctor, he brought the CDC recommendations on treatment. He showed it, because he's some around here, Pete doctors don't say that often. He's like, oh, it's Lyme disease. I need to look up what the treatment is. And Matt was like, here are the CDC's recommendations. We had it with them. All right. So that was disease of the day, and now we're going to talk about biochemistry. Did I make a mistake?
I'm just trying to make sure I understand.
Oh, that's probably me. What did I do wrong?
The way you
Oh, here. Yeah, that's an error.
Okay,
That may have been an autocorrect error.
sure.
But I'll fix it. I'm glad you saw it. It should not be capitalized. Good job. I was testing. No, I wasn't. It's a mistake. All right. I'm trying to do this. love chemistry. Love it. Oh, number 2. A lot of people sort of feel like chemistry was something they had to survive, right? To get to, it's a prereq, so you have to take it. And a lot of times when you take chemistry, you spend a lot of time doing inorganic chemistry. So I was joking earlier about the electrons filling the orbitals, and you have to memorize all of that and have bonding and things like that. And actually, the part of chemistry that I need you to know coming in here is the biochemistry. And sometimes they just do that on the last day. They rush through it at the very end. I hope that wasn't your experience, but what I want to do is just scratch the surface of the most important parts to micro. Because what I actually care that you understand is the big macromolecules. I can't reteach all of chemistry in half an hour. And I'm not going to try, but I am going to, this is way more about me reminding you what you need to know than it is about going into detail about each slide. Does this make sense? If you're feeling like, wow, I just don't remember any of this, then you want to read the chapter, come see me. There I can try to find some other activities online that you can kind of practice learning about these molecules. So this is way more just me pointing out you're going to need this for micro, you're going to need this for micro, than trying to recapitulate your chemistry class. All right, so I assume, of course, that you know all about protons, neutrons, electrons, and atoms, the different kinds of bonds, positively and negatively charged ions. You don't really need to read the periodic table, but you should have learned how to do that. Hydrogen bonds, electronegativity, basic ideas around chemical reactions. You should have learned about molecular weight, moles, pH, things like that. All of those are useful in biology. So in cells, we don't care about all the different kinds of chemical reactions. We only care about the ones that happen in cells. And so the major categories in the environment of a cell are chemical reactions that build things up. So these are synthesis reactions or anabolic reactions. And we care about reactions that break things down. So decomposition reactions, or sometimes we refer to this as catabolism. So this would be where you eat spaghetti and your body breaks it down and gets energy out of it. It'd be a catabolic process where you break down the starch into glucose, and then you break down the glucose and generate ATP out of it. Yes? Sound familiar? I'm going to open this with the quieter out here right now. Okay. Usually, when you're building something, it takes energy. So a synthesis reaction usually requires energy, right? If you're breaking something down, as you break down the chemical bonds, usually in cells, there's a release of energy. And that release of energy is usually tied to doing something the cell needs to do. Build something else, energy for movement, etc. All right, so here's just sort of a picture here of breaking down a big molecule into small parts and getting energy versus taking small parts, using energy to build a big molecule. Anabolism and catabolism. All right, so here we go, just to put the ideas for practice here. A person eats more food than they need in the form of starch. The body converts some of the excess glucose to glycogen to be stored in the liver. Does this process require energy or yield energy? What do you think? They're going to get a little bit of energy, but there's excess, right? And so the excess glucose is going to have to be built into big molecules of glycogen. So that process will require energy. They're going to get some energy from the food they ate too, right? Does this make sense? If you take glucose and you make glycogen, that's going to require energy. All right. You can't talk about biology and chemistry and the importance without talking about the importance of water. Because the chemistry of life happens in water. In chemistry, when you think about a solution that a chemical reaction occurs in, it's called a solvent, right? So that you've got the solvent, all the molecules are dissolved in and they're reacting with each other if they're reacting. The chemistry of life happens in water. So water it's your solvent. It's what your cells have water in them. And the molecules that are reacting in your cells, oftentimes those reactions are happening in water, or maybe there's a lipid-water interface where they're happening. Water is not an organic molecule, it's an inorganic molecule, and it's very polar. It's got some unique characteristics. So hopefully you learn in chemistry that water has an oxygen and 2 hydrogens, And that oxygen is not, it failed kindergarten, right? It doesn't share very well. Do you remember that? So it likes to keep all the electrons next to it, and it doesn't share nicely with hydrogen. Sound familiar? So that means oxygen has a slightly negative charge, and hydrogen ends up, not without its fair share of electrons, it has a slightly positive charge. So we talk about dipoles in chemistry, but it really just means that there's sort of a positively charged end to a water molecule and a negatively charged end to a water molecule. And this ends up being really important because this allows water molecules to hydrogen bond with each other. Remember hydrogen bonding? Okay. So that means that negatively charged or partially negatively charged oxygen, they're going to be attracted to positively charged hydrogen. That's called an electrostatic attraction, where the positive charge and the negative charge are attracted to each other. And you can kind of think of water molecules like Velcro, right? They like to stick together. If you take a droplet of water and you put it on a surface, it tends to bead up, right, and form a ball. If you did the same thing with alcohol or with gasoline, what would happen? Or oil, what does it do? Spreads out, because those molecules can't hydrogen bond with each other. They don't stick together like Velcro. So they're just going to spread out on the surface. Water likes to stay together. It's sticking to each other because of hydrogen bonding. Okay. What kind of bonds do we find between oxygen and hydrogen in a water molecule? Now, I want to be clear before I look at your answers. I'm talking about one water molecule, not two. One water molecule between hydrogen and oxygen in a single water molecule. And while you contemplate that, why don't I make this a better question?
Go find it.
Let's see. In a single, there. That's better. Did some of you change your answer? No? Let's see what you said. Okay. So if it's in a single water molecule where you have an oxygen bonded to two hydrogens, those are covalent bonds, right? You see where we're going with that? Yeah. Yes. All right. How about when we are thinking about between two water molecules? When you have it. Shh, don't tell them. Go ahead and answer that one. So this is really just to remind me of the different kinds of bonds, right? All right. All right, so I think those of you who missed the first one now got it on the second, right? The difference here? Hopefully that was helpful. All right, so just here's some pictures of hydrogen bonding. So we have our hydrogen with a partial positive charge, that's delta plus, means partial positive, interacting with oxygen that has a delta minus, and giving us that Velcro effect. So water